Hello everyone, my name is Ramesh and I am a research fellow under Professor Winkin Song in School of Computing. Today I will be talking about how to detect transpositions from DNA sequencing data. First of all, we all know that we have a genome and that's unique to each of us. However, surprisingly, if we take two random human beings, these will share the vast majority of their genome. Current estimates say that they will share up to 99.9% of it. And despite this, we are all very different. We all look differently, we all behave differently, we have different talents, we may have different diseases. And this is due to variations that occur in the remaining 0.1% of each genome. There are many reasons why detecting these variations is important. One of them is that they can be used in what we call genome-wide association studies, or GWAS. In GWAS, we study a large number of individuals, and we, first of all, detect the variations for each of them. And after that, we try to correlate these variations with specific traits that we are interested in. For example, in the case of cancer, we may be interested in knowing which mutations will protect us, or which mutations will predispose us to cancer, which mutations will cause cancer. And there are many different types of variations. They are usually classified based on size. Large-scale mutations are called structural variations, and due to their size, they account for the most differences between individual genomes. They are also responsible for many devastating genetic diseases, such as leukemia. And in this talk, I will focus on one type of SV called transpositions. So in simple terms, when a transposition happens, one piece of genome moves from one location to another. And the second location may possibly be very distant from the first. In this example, for instance, I have this, I've drawn this red piece of genome in location A. And what happens is this piece moves and inserts itself into location B. And what we obtain is a modified version of location B, which will contain the new sequence. As I said before, we are interested in detecting these variations. In particular, here we want to detect the transpositions. And in a typical scenario, we will have a reference genome, for example, the human reference genome, which is a complete or almost complete reference genome, which is assumed not to contain any variation. And we also have the DNA sequencing of a sample, for example, of a person. And we feed this data to an algorithm, which will predict a set of transpositions that hopefully appear in this person's genome. And there are many types, many platforms for DNA sequencing nowadays, but by far, the most used is next generation sequencing, in particular, Illumina platforms. And what NGS sequencers do is they are unable to read very long strings of DNA. So they will take each chromosome or each large piece of DNA that they want to sequence, and they will cut it down into very small fragments. And after that, they will be able to read each fragment individually by producing a very small read from each end of each fragment. So you can see here, we have a few fragments in black, and for each fragment, the machine will produce a read from the left end and a read from the right end. A read is a very small piece of DNA, normally around 100, 200, maybe 300 base pairs. At the end of the sequencing process, what the machine outputs, it's just a pairs of reads. So every information about the original chromosomes or even just the fragments that produce the reads is lost. And what we are left with is just these pairs, a very large number of them. When we sequence a human genome, we often end up with hundreds of millions or even billions of these pairs, but that's it. And the reason why Illumina NGS sequencing is so used today is mostly due to its cost. It's cheap, and it can be used to sequence thousands or even tens of thousands of individuals relatively cheaply. And for this reason, there is vast amounts of data online, publicly available. It's robust. It produces high quality data with very few errors. However, one challenge is that, as I said, the reads that are produced are very short. And this makes finding long variations very difficult, very challenging. As you can see here, what I did here is I took 5 publicly available human datasets, and I used 3 widely used, 3 of the most famous SV callers. And as you can see, none of them really has good performance in calling transpositions. Sensitivity or recall is less than 25% for each caller in all samples. That means that they miss 3/4 or more of existing transpositions. And the precision is also very low, which means that not only these tools will miss most existing transpositions, but they will also predict a very large number of transpositions that do not actually exist. And this makes downstream analysis of the data extremely confusing. So what we did is we identified the shortcomings in current approaches, essentially why existing methods fail. And with this knowledge, we developed a new algorithm, and which we implemented in a software package called Transsurveyor. And finally, we demonstrate its performance compared to the state of the art. So how do existing methods detect transpositions? So remember from our example here that we have one piece of DNA, this red piece of the genome that moves from location A and inserts into location B. And because of this, we obtain a new version of location B, which contains the red region inside. So in our sample, the transposition happened. So obviously the sample will contain the new version of location B, the one with the red region inserted. So when we sequence the pairs from it, hopefully we will obtain some pairs such that one read was sequenced from the green region and the other read was sequenced from the red region. After sequencing the pairs, the second step is called mapping. So mapping is essentially means that we want to find for each read, we want to guess which location of the genome it was sequenced from. And in order to do this, what we do is for each read, we find the most similar location on the reference. And the reference doesn't contain the transposition, so obviously the red region will be in its original location, location A. For this reason, when we map these two pairs, what will happen is one read will map to the green region here, but the other read will map to the red region in location A. And because of the nature of NGS sequencing, we do expect that pairs actually map very close to each other, on the same location and close to each other. However, as you can see, these two pairs do not. The two reads map very far from each other. We call this kind of pairs discordant pairs, and discordant pairs may be evidence for a transposition. If a tool, if a transposition as an SV caller finds enough discordant pairs, enough evidence for a transposition, then it will call the transposition. So why do existing algorithms fail? What we noticed is that we could find many groups of discordant pairs, like this one, such that one end could be clustered. So for example, here we can cluster these reads on chromosome 1, but the other end could not be clustered. As you can see here, these 3 reads, they all map to 3 different locations, even 3 different chromosomes. And so therefore, when the methods want to cluster the evidence and they want to find the actual transpositions, they are confused because they either predict 3 different transpositions here, or they don't predict any. And the reason why this happens, we found, is mostly due to mobile elements, which means that essentially the read sequences that jump around the genome are not unique. They appear in many copies throughout the reference, and this is a problem. For example, in this case, we have one transposition from chromosome 2 into chromosome 1. The sequence here being transposed is the blue and red sequence. However, as you can see here, in chromosome 11, there is a second copy, an identical copy of this blue region. And in chromosome 7, there is a copy of the whole transposed sequence. However, there are some small differences in the blue region. Now, when we sequence our sample, we obtain 3 pairs that are supposed to support our transposition. That means that these 2 reads here were also sequenced from chromosome 2. However, because this gray read was sequenced from the blue region here, when we map it, we have 2 equally good locations where to place it. So we don't know if we want to place it on chromosome 2, or we don't know if we want to place it on chromosome 11. And the same is true for the green read. Because this green read was sequenced from this red region, when we map it, we find 2 equally good locations where we could place it. We could place it in chromosome 2, or we could place it in chromosome 7. And what mappers do in these situations is usually they choose one location at random. If we are unlucky, in this case, they will choose chromosome 11 for the gray read and chromosome 7 for the green read. So what we did is we came up with this rather simple technique called End Remapping. And the idea behind it is to correct these wrong mappings, essentially wrong mappings of the gray and the green reads, and to recognize that the actual location should be on chromosome 2. So we want to actually remap the gray read onto chromosome 2 here, and the green read onto chromosome 2 here. And the way we do it is we use the stable end, we use the end that can be clustered in order to cluster these discordant pairs and say, okay, probably these pairs may support the same transposition. And after that, we want to determine which one of these 3 locations is the best candidate for the transposition. And in order to do this, what we do is we extract the region around these reads. So we obtain one candidate region from chromosome 2 around the black read, we obtain a second candidate region around the gray read on chromosome 11 here, and finally a third region from chromosome 7. And then for every candidate region and every read, we perform a Smith-Waterman local alignment. And as you can see here, because this is the correct region, chromosome 2, the 3 reads will align very well to this region, will align essentially perfectly. However, in the other 2 regions, because they are incorrect, the reads will not align as well. Either they will not align at all, or they will not align so well. And because of this, we understand that chromosome 2 is the correct location for the deletion. So we remap all the reads onto chromosome 2. And finally, it's easy to actually cluster the evidence into a single transposition, which is finally correct. And you can see that just by using this trick, we can improve the performance of transposition COLI by a substantial amount. So our sensitivity is much better than the other tools, is more than double. And we predict actually more than half of the existing events. And the same is true for precision. We vastly improve the precision of transposition COLI. And this gives us a much higher F1 score. If I'm not mistaken, it's around 3 to 4 times higher compared to the other tools, which is indeed a significant advantage. Thank you. If you have any question, let me know.